All right, so welcome back for the second lecture by Professor Roop Mallik, and today he is going to. So yesterday he talked about motors and membranes, and today he is going to talk about motors and tweezers.
Okay, good morning, everybody. So we today's talk might have a slightly different flavor with some things similar to yesterday. But Ambarish told me that in the spirit of a school, this should be a more introductory sort of talk. And until now, there has really been no talk on kind of trying to understand the way you do these kind of biophysical experiments on motor proteins. So that will be my purpose, to kind of introduce you to motor proteins in general, what they do. To some extent, I won't go into very many detailed biochemical cycles of that, and to some extent try to understand you why this is a good marriage between motor proteins and tweezers, why these experiments work very nicely when they do work. Very quickly, this is not necessary. This you people have gone through, and if this is a typical cell, then there will be cytoskeletal elements. Only the microtubules are shown here. There are, of course, actin intermediate filaments which are not depicted in this schematic, and there are many kinds of organelles which don't just sit around, which are transported along these secretory vesicles, pigment granules. Even DNA as a whole can be, partitioned, as you know, when cells divide. Bacteria, virus. So all this is very important for understanding pathogen infection also. And these kinds of cargoes can be transported in various directions along these microtubules. And as you saw from Sandhya's talk yesterday, when you look, let's say, for example, into axons, this transport is often happening in a back and forth manner, and you can get stuck at places and maybe come unstuck, or not come unstuck, remain stuck. So there are these tiny motors, and this picture is a little misleading again because they show single motors on this. That's usually not the case very often, and so they are transporting these cargoes. And here is, for example, what we have done in this fluorescence image. Here is the scale bar. That we have taken a kind of a macrophage cell. Okay, it's a part of your innate immunity is imparted to you because of these macrophage cells. And you can see, for example, these are the microtubules here shown in red, and these are small beads which are fluorescent, which have been eaten up by these cells, just like these cells would eat up bacteria, for example. Okay, and those can get transported. If this was a movie, you would see them moving around, but this is just a image. And yeah, so microtubules, nucleus, cargo in different colors. I was talking about cell division, and here again, that is, of course, a different kind of motor protein, but you will see that the organization of microtubules can be dramatically different when cells divide. And this different organization, you can see that there are microtubules between these two foci, and they will help you segregate the DNA because of a very concerted efforts of many kinds of motors and the dynamics of these microtubules. This is going to be a microtubule-centric talk. That's just because of my background. Of course, there is equally important and exciting things happening with actomyosin motors, and I apologize that I'm not going to get into that. Okay, this is how these motors look like under the electron microscope, just to kind of give you an idea of the scales. And this is a nice picture as you would draw of this same kinesin motor. And here is a dynein motor. This we should run. It is not over. Yeah. And here is another version. It is the dynein motor. These names are derived from the units of force, and kain is motion in Greek. And just again to remind you, the size scales are, this thing, for example, is about 10 to 13 nanometers. So this whole motor would be of the order of 100 nanometers. And being that size, 100 nanometers or so, they can carry cargo which are much larger than their own size. Like you know that mitochondria can be 2 to 3 microns in size, so 10 times more. And many of these motors clustered together, they can drive segregation of DNA, they can drive my hands moving around and my vocal cords oscillating. So all of this. So an important problem is to try and understand how force generated by these kind of molecules at the level of 10, 100 nanometers is amplified to what you can see visibly, okay? But that's not something we will really go into right now. Some numbers are important to keep in mind, as I was just mentioning. I mean, the dimensions of many types, of course, there is going to be a lot of variation in this, like neurons can be of the order of meters also, but most of the other cells can be 10, 20, 30, 40 microns of that order. The motors are of the order of 50 to 100 nanometers in size, depending on which one you're talking about. The cargoes can be 50 nanometer, 100 nanometer, like we were talking about synaptic vesicles yesterday. There can be few microns, or they can even be even larger than that, depending on what you're doing. Very important to remember that the speed of motion of a single motor is of the order of microns per second. Now, this is quite remarkable because if you scale it up, you will see that these things, the cycles which allow these motors to move, we will come to a little bit more of that, they must be really rapid for something which is, let's say, 50, 60 nanometer in size. To be able to move at 2-3 microns per second, they must go through enzymatic cycles really, really fast, okay? And that's, quite remarkable. Yeah, that's what I mean. You can have of the order of 100 cycles completed in 1 second, okay? When I walk, I don't complete 100 steps in 1 second, obviously, okay? So, what are these motors? They are essentially enzymes in which a chemical cycle is coupled to a mechanical cycle, okay? And the chemical cycle must therefore complete 100 times in a second and drive a mechanical cycle an equivalent number of times. That's how you can get this kind of motion. What is the energy available? From 1 ATP is of the order of 25 times the thermal energy. So, so that's what can allow you to go significantly above the thermal floor and induce something which is meaningful. And this you can also express it in, because it's energy, you can express in force times distance. And these numbers may sound strange, but so this is piconewton into nanometer. I mean, normally when you talk about energy, you talk about Newton meter, but we are talking in terms of much smaller units because these are much smaller machines, okay? Not like you and me. For example, if I may ask you, so the work done that we would do typically in our lives would be of the order of Newton meters, right? We can pick up objects, we can apply forces of the order of newtons or tens of newtons over distances of meters, right? So when I'm moving this, I can, if I pick up a slightly heavy object, that's what I would do. Apply forces of the order of newtons or tens of newtons over distances of meters. So you can then calculate how much smaller objects we are talking about here. Because this is 10 raised to the power, this piconewton, and this is 10 raised to the power minus 9. Yeah, so the work done per cycle and how we calculate this, maybe we will come to that later, is of the order of 50 piconewton per nanometer. Again, this will be different for different kinds of motors. So if you assume that you are using 1 ATP molecule per cycle, your efficiency would be of the order of 50%. Again, this is for a few types of motors, this number can change, okay? So, and in fact, we are talking about linear motors. There are motors which are also rotary motors, okay? For example, which use ATP to drive motion. And for those motors, people have calculated so this would be 50% efficient approximately, okay, right? But for those kind of rotary motors, people have calculated efficiencies of the order of 80-90%, which is, really amazing. Remember that for a 50 nanometer size object, let's say a synaptic vesicle in some watery medium of viscosity close to water or a little more than that, this would be your diffusion constant like. Okay, so and for larger objects, this number would really drop rapidly. Okay, so that's why you need this kind of active motion. If you have to traverse the cell, a typical cell which is 10, 20, 30 microns long over a period of seconds or minutes, you can't do it by diffusion if you are kind of large object. Another important thing, we won't go into too much of this, is duty ratio. Okay, so one way to understand it is that so this is basically these are cyclic machines, right? So part of the time, so suppose I'm walking like this, okay, then I'm always one leg or the other is attached to the ground. So my duty, so my duty ratio for each leg is 50%. For the total machine is 100%. Okay, something is attached all the time when I'm walking. But when I'm running, that is not true, right? Because when I'm running, there will always be instants when both legs are above the ground. Okay. Now, for these kind of machines, kinesin, dyneins, they don't run, they walk. Okay, because if they ran, there would be an instant in the cycle when both legs would come off the substrate, let's say the microtubule, and if that happened, the diffusion is so rapid here, particularly for an object of 100 nanometer, they would just be swept away. Okay, so as long as they are moving, they are walking, they are not running. I hope you understand the difference between these two. Okay. Yeah, so that's the duty ratio, and this duty ratio for kinesin for a given head is close to 0.5, okay, which means that it is always attached. But there are other motors which, for example, some motors which function in mitosis, and Ambarish can tell us more about that, which have lower duty ratios. For example, myosin, muscle myosin, has a duty ratio which is significantly less than 0.5. It is about 0.1 or something like that, right? Yeah. So you have a filament, can be microtubule or actin, and you have the legs of the motor, and then this is moving here. And if you look at this region where a protein surface is interacting with another protein surface, and let us say there are some charges within these proteins. Okay, for example, that tubulin has a charge. Let's say the sole of the motor also has a certain charge distribution, and there can be other factors also. You would form a potential, and energy surface here, right? And because of a charge-charge interaction, and in some models, people think that this interaction between these two surfaces leads to a potential energy surface which is, sorry, I have not drawn it very properly. It's not symmetric. Okay, so this is the situation when this guy meets this guy, and it has to walk across this. Okay, so it's an asymmetric energy surface. But when these two are not close to each other, this surface looks flat, obviously, because the charges are far away from each other and they are screened by whatever is around. Okay, so the job of ATP is to switch this system from between these two states. Okay, that's called the flashing ratchet model. You switch the system between these two states, and when you go to this state, which is when you are bound, that is when the interaction is happening, that's why you see a potential energy surface. If you are, if you are here, you start diffusing. Why are you diffusing? So you have to imagine that one head is bound and the other head is diffusing like this. And why are you diffusing? Because of thermal fluctuations. And you might, in a given time, you could cross over this barrier and come to the next site. But because of the asymmetric nature of this, it is highly unlikely that you will go back like this. Okay, so you'll come here and then you will diffuse around here, and then it's more likely. So the nature of this surface gives rise to an asymmetric potential, energy surface, and because of that, when you combine it with the ratchet-like behavior driven by Brownian motion, you get motion largely in one direction. There are some back steps and all that, but let's not get into those complications. Okay, so yeah, right. So, well, that's that for motors. If there are any questions until this stage about these details, I can try to answer. Then I will slightly change what I'm talking about, and then we will come back to these motors later again. Yeah. What is what? No, this is not protein. This is apostate. Apostate when there is no nucleotide bound. That's called the apostate. ADP PI is you bound ATP, you hydrolyze the phosphate, the terminal phosphate, but the phosphate has not left yet. So you are in an ADP and phosphate are both there. So that's. It is strongly bound, yes. This is for this motor. It need not be true for all motors. And apostate is also strongly bound. Okay, and then when you release the phosphate from here eventually, okay, then you will go to a weakly bound state where this leg will come off. Okay. So when this curve is going down, is it the time when the motor switches its leg? When this curve, no, this curve is in space, it is not in time. It is a static thing. See, it's not really static because you are fluctuating between these two potential surfaces. Okay, and when you are far away, when you have lifted your leg, then you are seeing this because you are far away from that surface. But when you have brought your leg closer again, then you will go here somewhere. No, so that's what I was saying. For example, for kinesin, for motors with a high duty ratio, there will be one leg bound, but the other leg is switching between these two states. Can I just add? Yeah, please. Go ahead.
This kind of model is generally used for the motors which are monomeric, just one head. Okay, this model is much.
So I'm showing you about one head here, essentially. Okay, so the other head is having a phase shifted cycle like this. Okay, so I'm not talking about that. We're talking about one.
Very accurate for motors which just have single head. This is a dimeric motor. So for them the things can be slightly.
So you, for the dimeric motor, you have another version of this going on, slightly delayed in time in the other head, but it may not be exactly similar. Yeah. Well, there's no answer to that. That's how it has evolved, to use, so that you develop some kind of polarity. Yeah.
Microtubule has an inbuilt polarity, and that is the reason for this.
Yeah. So the microtubule has a distribution of charges, right? Let us say, if you believe that this is for charges, and the surface which is coming in also has certain distribution. That gives rise to such a potential energy surface. Now, this is a model, okay? Now, I don't know how accurately that has been measured experimentally. That's something debatable. Yeah. Is there anything else? Okay. Okay, so now let's move on from a little bit from motors to the use of optical tweezers. And why we are doing this? Because many kinds of experiments, many of these things that we have learned that are shown here in this picture are actually derived using optical tweezer sort of experiments, okay, where you, and combined with structural biology, of course. So it's useful to understand how these optical tweezers work. So we have all seen, either on TV or in real, these kind of, comet tails. Now, do you people know why you have such comet tails? Somebody must know. There's no atmosphere in space, right? So it can't be that, there's like dust blowing here. It's not like that, okay? So this is because of the photons coming in from the sun, okay? In this case, depending on what it is going towards, okay? So you have, let's say the sun is here, okay, or some large star is here, which is emitting a lot of photons. Those photons come and hit this object, whatever is flying through the sky, and because of the momentum of those photons, so you have to imagine that, like this in the laser, okay, things are going to be shot off from this, okay. So in other words, I mean, all of you know this, that photons have momentum, but this is a one dramatic example of what photon momentum can do, okay. And that's, I mean, I've actually written it that, and this is the momentum of a photon, Planck's constant divided by lambda or f by c. And of course, if, I shine light on this gentleman, I can't blow his hat off, okay, because I won't have, the momentum of the photon is too small for an object of this size, okay, it will hardly make any difference. But if I am a bacteria, okay, or I am a micron-sized object, let's say like an organelle inside a cell, then it makes a difference, okay. Then you can use the momentum of the photon. I can't push my hand back because of this, but if I had a slightly more powerful laser and if I have a bacteria, I can push the bacteria away, okay. And that is the basic principle that is used in an optical tweezer, the force generated by light. And indeed, yeah, and there are actually spacecrafts which use this principle where you have this kind of a solar sail. People must have heard about it. You have a solar sail and you have a satellite, and if you orient it in the right direction, the photons from the sun, let's say, are coming and hitting it, and you will propel this object in one direction. Okay, so this is an example of radiation. Radiation. This is called radiation pressure. Okay, pressure because force because of the force per unit area because of the photons coming in, and this is an actual solar sail. Okay, so the photons come and hit from this side and this propelled. So the momentum of a single photon is this. You can look up your textbooks, 10 to the minus 27 kg meter per second. And if this is a typical laser pointer, you will be emitting something like this photons per second from this, okay, depending on the power of the laser. If this is a milliwatt laser, let us say, okay, you will be emitting so many photons per second. And if these photons stop dead as they are doing on my hand, okay, the change in momentum would be 10 to the power minus 12 newtons. You can calculate this easily. And that would be of the order of piconewtons. So if I take a more powerful laser, I can increase this because then I can increase the number of photons per second, okay. I basically have more photons coming in and hitting per unit time. And you can see that from this you get energy, a force of piconewtons here, okay. I assume, let's say of the order of picoseconds. Okay. There is no justification. I mean, it's an instantaneous stop. Because we know, no, so in a way, the justification is little bit, putting the horse before the cart, because I know that the kind of forces you generate are piconewtons. That I can measure. So I would assume, therefore, that if you put in this kind of a time, you would get something like this. Okay? It's the other way. Yeah. All right. Okay, so why are we talking about all this in the context of optical tweezers is because what happens in an optical tweezer that you have an object, let's say it can be a plastic bead or it can be a bacteria or a mitochondria, something that changes the direction of the incoming photons. It refracts. It's a refractile object. Okay, if it refracts, it means it bends light. So if I send in a photon like this, and if I can take the snapshots of the photon as it goes through this, not easy to do, but you would see that it comes in like this. when it goes from refractive medium of one refractive index to medium of another refractive index, it has changed its direction. You can see that there is one change here. And then when it emerges from the other side, it is again going from one refractive index to another refractive index. So it is again changing direction. Okay. And so you can therefore, we will do that in the next slide. This change in direction twice here leads to a net force on this on this particle in this direction from the light that is coming in. Okay. And in reality, you don't have a single photon, because if I'm shining this laser through this, you have a stream of photons. So this is what is happening. Okay. And when this object is at the center of this stream of photons, okay, then there is no net force. It sits stably. Okay, that's why it's a trap. But as soon as I move this object out, there is a restoring force. We will come to that in a minute. Okay, and that restoring force is, that's how, because let's say there are thermal fluctuations. It is pushed out a little bit on one side, it'll come back, okay, within certain limits. Or if there is somebody pulling it out, let's say a motor protein, okay, then you will you can apply a restoring force against that motor protein. And so let us go into that a little bit. So Arthur Ashkin, who got the Nobel Prize in Physics of some 3-4 years ago, is credited with the discovery and the implementation of the optical tweezers. So what he did is that he took laser light coming in from this side here, okay, and this is like, imagine a solution, okay, let's say in water. He had some small particles, okay. This could even be, for example, pollen grains, okay, they are of that size. And he took actually small plastic beads and of this kind of mass, and he calculated that if the mass is 10 to the power minus 15 kg, depending on the density of the size and the volume, etc., you should be able to exert a force on this, okay. And so that's what he saw, that when you shine a laser on this, then it pushes this bead to one side. That is essentially this, the photons are coming and hitting it, okay, momentum of the photon, and all these beads go and accumulate on the other side, okay, because they get, they have nowhere else to go after this. Then what he did is that he took another laser. Where is the other laser? Yeah, so these accumulate on one side like this, and if you have another laser coming from the other side, and you can align these very nicely and match their intensity and all that, then you can trap all these objects in the center, okay. And so that was kind of expected, okay. But the unexpected part was that if there were beads floating around outside this focal volume in an orthogonal direction, so this is the direction of propagation of the laser, okay, and it's a sort of a focus, it's a focused laser beam. Sorry, I forgot to say that. You can see that it's a focused laser beam, okay. The beads which are away from this are started to move in towards this clump, okay, in an orthogonal direction, which was unexpected because if I'm hitting it like this, how am I generating a force in the perpendicular direction? So that is when, people thought about this, and they realized that not only, so this nomenclature is important, there is a scattering force, okay, we call it F scatter, which is expected because if I come and hit you, then you will get pushed away. But there is also something called a gradient force on these objects, which arises because of the non-uniform distribution of light along this axis. There's more light in the center, there's less light here. So there's a gradient of light, okay. So how does that work? We will see that shortly. But And this had huge applications. In fact, look at the number of papers that Ashkin published over the years. And he was having a good time, clearly. But many of these applications were based, when this initially started, were based on, understanding levitation of atoms. That's how people made Bose-Einstein condensates. And there's a very strong applications in physics, and that's where Arthur Ashkin was also coming in from. But essentially this did not go outside this kind of fields, because in many labs, to make an instrument where you have these two lasers perfectly aligned, it was not easy to do that, okay, and perfectly matched in intensity. But then what people realized after a while, that you actually don't need the second laser, okay. If you just have one laser, you can do this with a single laser, okay? And that's what we will look at. And when that happened, then this really, that simplified the whole experimental setup, and people could just take a microscope, shoot in one laser from one end, and put in a few lenses and mirrors, and you could get an optical trap. And that's exactly what we have done, okay? Now, apart from that, why this caught the interest of biologists really was because Ashkin showed in this paper in 1987 that you could trap single yeast cells, okay, in the optical trap and keep them trapped for a long time, and they would divide, okay. Now, obviously, if you are reproducing, then you are happy, okay. That's the assumption in biology. So then it means that the optical trap, if you obviously, keep in mind the certain simple things, and you can work on biological systems also. You are not frying them up, okay. So this really, all these kind of developments, see the division of yeast cells in infrared. IR means infrared. Why? Because the wavelength used in these optical tweezers is normally the infrared region, okay. And this was the kind of power he used. And so let us see how it actually works very quickly. I will go through this. So before that, there are a lot of possibilities in biology, and many of them have actually been realized. So apart from a magnetic, using a magnet, I can't think of any other way in which you can exert force at a distance, okay. But unfortunately, most things inside cells or inside your body are not magnetic, okay. So it's not easy to exert magnetic force in biological systems, but many of these organelles, cells, they are refractile, okay. So optical trap you can use not just in physics, but you can also use optical traps inside cells, and that's something which we have also contributed to, and I will talk about that tomorrow, okay. You can measure the mechanical properties of cells, you can stretch them, you can measure mechanical properties of membranes, protein filaments. Of course, all of these has its own, ways of doing these experiments, which I'm not going into. By using more complicated versions of optical traps, where you have multiple optical traps, each of which you can manipulate in time, you can build biological structures. You can take 7 organelles of different types, bring them together in a very controlled manner, and see if there is membrane fusion. Yesterday we were talking about membrane fusion, for example, okay. So you can engineer all these kinds of events using these kind of assays. It has been used for in vitro fertilization, okay. You can imagine that you catch the egg and the sperm and you can do in vitro fertilization. Of course, single molecule mechanochemistry of enzymes, which has been the most celebrated use of these optical tweezers. But let's go a little bit into trying to understand why why it works, okay. So what happens is that in an optical tweezer, I take a laser like this and I put a lens above that, okay. When I put a lens, this light beam, it's assuming that it is coming out parallel from here, will get focused at certain distance, which is the focal length of this lens. That lens typically is the objective lens of a microscope, okay, which has a very short focal length, particularly if it's a high NA objective, it has a short focal length, high magnification objective. So this distance where it gets focused would be less than a millimeter or something, okay. Now it gets focused somewhere, and then let's say you have an micron-sized object. Okay, let's say a plastic bead. Let's assume it's spherical, just to keep it simple. Above that, and this has a higher, the refractive index is higher than this, suppose, okay, which is typically the case, okay, because this is water and this is, let's say, some glass or silica, then the light would bend in this manner, okay, away from the normal towards the normal. All of you know that. I'm not going to go through that. And then when it comes out, it will again bend away from the normal, okay. Now, so when this happens, you can clearly see that if this light is coming in and hitting it, there is going to be a component of this along this direction, right? So you are going to push this object upwards. That is the scatter force that I was talking about earlier, okay? To some extent, this will be balanced by gravitational force going downwards, but not completely, okay? Now, so that is the scattering force. But there is something more, which is that if I now draw the vector diagram, okay, so P out, this is the direction, P in, this is the direction, and the net change in momentum will be, if you just draw the vector diagram, it's going to be in this direction, okay. So this is the change in momentum of the photon, okay. So there's going to be a force, corresponding force applied on this object, which is going to be in this direction, right? So that's what this force is, okay? Because of the passage of a single photon, there is going to be a little kick on this object in this direction, okay? This little kick can be resolved into two components. This component will, to some extent, balance out the scatter force, right? And this component is in this direction. Similarly, because of the other beam, which is on the other side, there's always going to be a symmetric beam. You will have this, okay? So what you have ended up doing is that if you go along the circumference of this, all of the horizontal components will cancel out, okay, assuming that it is in the center, but all of these components will add up. So what you have done is that you have balanced the scatter force by a downward force, okay, and therefore you have trapped this object in the vertical direction, correct? But you have not trapped it in the horizontal direction yet, okay, in this plane, because it can always diffuse away, because remember, this is all happening at, room temperature, so there's always going to be thermal diffusion. You can actually see that. So now I have trapped it in one dimension, which is along the direction of the light. How do you trap it in the orthogonal plane? To understand that, let us just look at this, okay? So suppose I come in and pull this object on this side, okay? I come in, or thermal forces can also do that. Let's say it gets pulled onto one random side, then the distribution is not symmetric anymore. Okay, remember that the light that is coming from here is highly focused. So as you go away from the center, there is less light. There's a gradient of light. Okay, so now a large part of this object has gone away from the light. So the number of photons passing per unit time through this side is going to be less compared to this side. Okay, therefore the time integrated force over whatever reasonable time is going to be more on this side, right? Because there's just more photons going through that side. It's closer to the center, and therefore you will have an asymmetry. This component will increase as compared to this component, so you will pull it back. Okay, and this happens in all directions on this plane. So that is how essentially you trap it in 3 dimensions. Is there any question at this stage, or should I move on? Okay. All right. Was there something I'm forgetting? Yeah. There are other ways of explaining this. This was the ray optics regime where, depending on the relative dimension between the wavelength of the light or the object that you are trapping. And there are other regimes where you can explain dipole, but I will not get into that right now because of lack of time. So how do you actually make such an optical trap? So the simplest version that we have in our lab is like this. What you do, you take a microscope, you almost always you will build an optical trap on an inverted microscope. An inverted microscope is where your objective is down here and you're looking up at the sample, okay? An upright microscope is the other way. Do people know why inverted microscopes are used versus upright microscopes? Who knows? What is the advantage of an inverted microscope? Yes, that's one advantage, because if your objective was like this, and then if you're looking from near the objective side, then you have to keep your cells, if you're keeping it in a liquid, gravity works only downwards. Okay, so so that's one advantage. You can do live cell imaging. Now, for optical tweezers, the main reason why you do this is because you the laser beam that is coming in is below eye level. Okay, if I had an upright microscope, the laser beam would be coming in here, that would be at your eye level. That's extremely dangerous. Okay, because this laser is infrared, you will actually not even see it. So you will not know, but you will go blind if it goes to your eyes. So therefore, one has to be a little careful. Okay, so what you do is that you take a laser, a stronger version of this one, and this laser is such that if you look at the beam cross section, it is called the TEM00 mode, okay, where the distribution is Gaussian as it is propagating. If I cut it like this, the distribution is the most light at the center and going down at the side. There are different versions. There are Bessel beams, other kinds of beams also people use for optical tweezers, but I will not get into that. Those are used to impart angular momentum to objects also, okay, to rotate objects. But yeah, so you take a laser, you have various mirrors. What you need to do is that typically the lasers you will get will have, the diameter of the laser will be maybe a millimeter or so, okay, 1 to 2 millimeters. You need to expand that laser beam, but still keep it collinear, okay? So that you do using a beam expander, which is simply a telescopic arrangement of two lenses. I will not go into that, but I hope you people will go back and take it as homework. Look at how you can make a simple beam expander using two lenses, okay? But the result of this is that if you send in a 1 millimeter beam, you will probably get a 4 millimeter beam, still collinear coming out from the other side. And then you have two more lenses. This is the simplest arrangement. And in these two lenses, they are both of focal length some f, let's say, and they're placed at 2f distance apart. And if I now change the distance between these two lenses, I can make this beam on the right-hand side either slightly divergent or slightly convergent, right? Depending on the distance between these two. You can draw it out and check for yourself. It's easy to do. And when I make this beam slightly divergent, then the place, so remember that this beam has to go in through another lens here, which is the objective lens. So there is another lens sitting here, which I'm not showing. It's inside this. If the beam is coming in slightly divergent and going in through that additional lens, it will focus farther up, right? If it is convergent coming into this beam, it will focus down. So by changing the distance between these two, I can, depending on where my sample is, I can move the focus of the optical trap up or down, right? So all this you do, and then you have to align all these mirrors and beams and everything. It's easier said than done, but once you have done it, then you should have a functional optical trap. So this is what like our trap in our lab looks like right now. Let me just quickly go through this. So I've taken this, I got on a stool and took it from the top just to kind of give you a picture. So here is the laser, okay? You can use a very fancy laser which costs a lot of money. We use a very simple one, which is a $700 diode laser. Okay, these are manufactured here also sometimes, and if it goes bad, we have been using this for almost 2006 to 2022. We have been using a single laser, okay, and it has been working fantastically for us. But if you use a complicated version, every few months you have to align it, realign it. So we did not have the need for it, so we never did it. So you have this is an optical fiber, okay. This is called a single mode optical fiber. where it filters out all the other modes and lets only the TEM00 mode propagate. Okay, so you get a Gaussian beam. So you automatically filter, ensure that it is a Gaussian beam. This light goes in at the back of this. You can see a mirror here. If you look carefully, there's a mirror here. Okay, that mirror can be adjusted like this. And so that mirror sends this laser beam here into the back of this microscope. Inside this microscope, somewhere here, is a mirror which sends the beam up into your objective lens, okay? And then the light continues through the objective lens and gets focused here. That is where we keep the sample. Then that light beam continues upwards. After passing through the object that you have trapped, it continues upwards. It diverges, remember, right? Because it is passing through this objective lens, it focuses and then it diverges. And you will not see there is a mirror here, a dichroic mirror inside this at a 45-degree angle. That dichroic mirror sends the light this way. There is another lens here which focuses that diverging beam and kind of makes it, a little less diverged. And then this light beam comes and hits this here object here. It is called a quadrant photodetector. Okay, so there are 4 photodiodes on that, and I will come to that in a little bit. And why are we doing all this? Okay, I forgot to say, we are doing all this because suppose I have trapped something right here, okay, and that trapped object, let's say a bacteria or something else, is moving in this optical tweezer. So that object is working like a lens. Another lens, right? It's a spherical object, let's say, and the light is passing through it. It is moving, it is like a lens which is moving. When the lens moves, the place where this light will come out will also move, right? When that happens, this laser beam, what is focused on this detector, that point position will change, okay? And I can easily measure how much this object has moved by measuring the displacement on this detector here. Okay, and I can do this at very, very high sampling rates, megahertz rates. Okay, so I can sample the displacement of an object every microsecond by doing this. And I need to do that because I had told you that these enzymes take hundreds cycles in a second. Okay, so that's why I want this kind of a time resolution. Okay, so that was the simplest optical trap, but not all optical traps are that simple. This is one version in Matthew Lang's lab. So what Matt has done is that he has combined single molecule fluorescence with optical tweezers, positioning various kinds of acousto-optic detectors. And I mean, if you are interested, you can read about it. This is a fantastic instrument, but not something that we have. It's useful to just for you people to, there are certainly more laboratories which I am not aware of or I have not listed. Some people in our country who work who use optical tweezers for various kinds of problems that they address. Some of them work on biological problems, some of them work on more physics-oriented problems, but it's useful to know who are the people. And we will also, in our own department, IIT-Bombay, thanks to Ambarish, we will also have a fairly advanced optical tweezer setup, and we hope that in next 6 to 7 months it will be operational. So if any of you have something in mind, please, So
we have a much more fancy version than this.
Correct.
Optical tweezer with a confocal microscope and a magnetic twister.
Yes. So you can do whatever you want there.
Yeah.
Okay, so if you get it all correctly aligned, and I hope this movie runs. This is what you see. Oh, didn't work. It's not working. Okay, sorry. Yeah, so this movie is not working. But if it, so what is you're essentially doing is that you are seeing the laser spot in your microscope. Okay, and this is the interference pattern. Okay, these are airy disks. Okay, so you see this concentric circles here, and if I move my objective up and down, you will see these circles growing smaller or larger, because you have to imagine it's like a hyperboloid like this, and you are cutting it depending on where you are moving this, and you will see the diameter of that. Okay, and Now, so why are we doing all this? Because we want to trap something. So, and let's see if that happens. I'm sure this movie will not run. Okay, sorry, but maybe I will go out of this and I can demonstrate to you, but yeah, it works. So anyway, so so to kind of connect back, how much time do I have now? 12:30? 12:30 I should end, right? Or I should end later?
12:15 you started.
Okay, so I have to 12:30. 12:30 is okay. 12:30. I can't speak for so long. My throat goes dry. Yeah. So we use these kind of optical tweezers to do many things, but one of the things that we do is to understand the force generated by these kind of proteins, okay, motor proteins, which are transporting a cargo, let's say, and the size is written here. And I have shown you this yesterday, and you can see that one cargo is these little pigment granules. Now, interestingly, you can't use optical tweezers with these pigment granules, okay? Some of our friends have tried. As soon as you focus the optical trap on one of these, it explodes, okay? Because it absorbs so much light. It's a pigment, right? So it absorbs light and it just simply explodes, okay? So we have at some point thought, so these are cells that you can extract from the scales under the scales of fish, actually, okay? So because fish can also change color depending on, what environment they are in. So these are called melanocytes, and you can extract them from fish scales. If you do it from normal fish, you have these pigment granules, but there are fish, let's say zebrafish or the kinds of fish which are called albino, okay? So they don't have this coloration, and if you, what we have been wanting to do for a long time, we just not got to it, extract these kind of cells from albino fish, and then if you shine the optical trap, you may not have this problem, okay? But, hazaron khwahishein aisi. So the first kind of measurements that were done were from the very celebrated work from the lab of Steve Block at Stanford, and also James Pudick in different kinds of motors, of course, they're talking about. And here is the paper from Arthur Ashkin, where they looked at the organelle transport measure in vivo using an infrared laser trap. But fortunately or unfortunately, I don't know, what happened over the course of years is that a lot of these kind of experiments got focused away from real biological systems, okay? Rather, people moved to isolating single motors, attaching them to very artificial sort of cargo, and doing very detailed measurements of these kind of motion in a very artificial environment. And we have learned a lot from that, but I think to some extent over the years, we have also missed out on how biology works. Of course, everybody has their own interests, and but that's what I feel. So I'll talk about that a little bit tomorrow. What we have tried to do is, try to go back to some of this kind of work and do these kind of measurements directly inside living cells. It's quite non-trivial to do that, and I will tell you what the problems are tomorrow if there is time. And what we have also done is that, coming back to what I started with, motor proteins and membranes, okay? Typically, these experiments that have been done, let's say by these gentlemen, are where they have taken an artificial cargo, let's say a plastic bead, which has no membrane on it, okay? And you attach the motor protein to it. Initially, it was done in a very random sort of way. Now people have better methods to do it, and look at this kind of transport, force generation, etc., etc. But there are, it is quite likely that you are not seeing what happens inside cells, and those are issues which are still under debate, and people are trying to address those things. Okay, I should have checked this, but anyway, so in this is another movie which will not work, but what we have done here is that we have purified tubulin from some animal source, in this case from the goat, brain of the goat, and you can polymerize the tubulin by giving it GTP. And if you look here very carefully, you will see a thin line here, okay? And that extends across this, so these beads are 1 micron, so that gives you an idea of the scale, okay? From here to here would be maybe 200 microns, 100 microns, something like that. And if I run this movie, which I can't right now, you will see that all of these are moving in one particular direction, okay? And that happens because we have also purified the kinesin motor from the same source, goat brain, and we have given a liberal dose of kinesin to these beads, and that results in the motion of these beads along these microtubules, but which I cannot show you right now because the movie is not working. Then what one can do, I think I had showed in the first slide, you can take one of these beads and shine the laser beam like this at this bead in an optical tweezer in a microscope. That's what I was talking to you a little while ago, and you will see something like this happen, okay? The motor will try to pull it out, and as it does this, remember there's a restoring force from the optical tweezer. This object will fall back, the motor will try again, it will fall back, and you can record these events really, really nicely. And to do that, this is so at last I have a movie that works, okay? So, because it's a GIF file. So, so this is the center of the optical tweezer, and you can see that this object is being put, this is a 1 micron size bead, and it is being pulled out. So there is a microtubule stuck underneath this, which you can't see in this picture, but, and there are motors also stuck to this, which you can't see again in this picture. And if you let it run, it goes out, comes back, goes out, comes back. You will see repeats of this event. So the force from the motor is in this direction. Why? Because this is a kinesin motor, and the microtubule which is sitting underneath it has its plus end on this side, okay? And you will see this, and the trap is pulling it back like this. So if I reduce the intensity of the laser or switch it off, it will just walk off like this, okay? Then, what happened? Yeah, then what you can, as I was explaining earlier, this laser beam gets focused and it's then incident on this kind of a detector, okay? This is a quadrant photodetector. It has 4 quadrants, each is a photodiode, and when light is incident, it generates current proportional to the number of photons, to the intensity of light coming onto it. And when this object moves like this, the shadow of this object also moves like this. If it is moving in a perpendicular, if the microtubule was oriented like this, then it would have moved like this. If the microtubule is oriented at a 45-degree angle, you would get a component along this and a component along this, okay? So you can, when you do these experiments, you can rotate your axis and have all the component along one direction, etc. I'm not going to those details. But the kind of data that you get from these is, you are essentially measuring this displacement, okay, as a function of time, and that is what is shown here. This is time, and typically this whole event will take maybe 1 or 2 seconds, okay? So I don't have the scales here, but this is roughly 2 to 3 seconds, okay? And on the on the y-axis, you have this displacement, we'll call it x here, and you see that this is the position of the bead, and the slope, this is because this distance, this is time, this slope is the velocity, right? So, and the slope reduces as you move away from the center of the trap. This is the center of the trap. This is the 0 position. This is this vertical line, okay? As you move away from the center of the trap, you reduce your velocity, and you ultimately at a certain point where you can't move any farther because the motor is now generating a certain force, it can't overcome the optical trap. In this kind of an optical tweezer, a simple one, the farther you go away, you have to imagine that you are pulling a spring, okay? And the force, the restoring force is proportional to this displacement. x, because it's like a linear harmonic spring, okay? So that's why I've shown this spring. If you have gone this far, then you have stretched the spring so much. And the force is the spring constant, which I will come to in a minute, into this distance. But if you have gone here, then you are exerting more force. You have stretched the spring more, right? So now what you can also, if you look carefully at these arrows, you can see these discrete events, okay? These are the individual steps of the motor, and you can see these steps here and not here for two reasons, okay? One is that it is just too fast to be detected here, okay? It is just going too fast. Second is that over here, you are at the end of a stiffer spring, so the thermal noise is less, okay? So you can resolve these steps now when you are, let's say, 50-60 nanometers away from the center of the optical trap. And by using these kinds of, getting this kind of data, you can really learn a lot about the mechanochemistry of these motor proteins. And some of these things that we have been trying to do, what we are trying to understand, I mean, maybe I will talk about that tomorrow. Yeah. Yeah, so this is what I was telling you. I think this slide is not necessary, but just to kind of remind you, the restoring force. Why is it important to know this restoring force? Because if I want to measure how strong you are, I have to do an arm wrestling with you, and I should know how strong I am, right? Otherwise, I can't. So I need to know how strong the optical tweezer is, okay, depending on the intensity of the laser. So I need to know this number, the spring constant, okay? It is Newton by force by distance units. And you can then simultaneously measure force and displacement. If I know this, I measure the displacement and I simply multiply it by the known spring constant, so I can also get the force. Okay, so I can simultaneously measure these two things, and they are both useful in understanding how single or groups of motors work. Yeah, so I think this is not necessary right now. I've already talked about this. And there are many different kinds of adaptations of these experiments which people have done. And typically, for example, here, if there is a protein, let's say, which binds DNA, okay? Let's say a protein which moves along the DNA helicase or a DNA-based motor, which moves along the DNA and let's say unwinds the DNA or does some other kind of modification to the DNA, then you can take that protein, okay, and you can bind it to the DNA by some chemistry which we will not get into right now, and the other end of the DNA, you can bind it to this kind of a bead. Always remember that typically for optical trap experiments, you need a large refractile object, otherwise you can't do anything, okay, because you need something to bend the light, and if it is too small, it will not work because it works on the principle of a gradient of light. If you are too small, then the gradient across that object will be too little, okay? If you are too large, then the thermal forces will be too large. So you have to be at a certain, size range. Typically 1 micron works very nicely, okay? Yeah, so you have this. Why am I going back all the time? Yeah. So what you can then do is that you can, for example, here is an experiment where you have RNA coming out of this. Okay, let's say it's RNA polymerase. Okay, it's making mRNA here, and you have RNA polymerase, RNA-P, which is bind to some kind of a chemical tag, HA, okay? And I have an antibody against HA, so I can stick the RNA polymerase to this cover slip, and then I give it a piece of DNA, and one end of the DNA, just like I showed you here, is bound to this bead, which is in an optical trap. Now, when the RNA polymerase gets to work, it is basically going to pull the DNA like this, okay? And going to read it and make the RNA, so you will have RNA coming out from one side, and you get data which looks like this, okay? You know the distance that you have moved. How? Because you simply measure the displacement of this. How? Assuming that this has, you have to correct for some compliances, but assuming that you have moved so much, you will move this bead out so much from here. If you have pulled 40 nucleotides, maybe the bead will move out 30 nanometer or something like that, okay? So from that you can, from the distance displacement of this bead from this center of the optical trap, you know how many nucleotides have been read through and you made mRNA, and you plot that as a function of time, and you can, various things which I'm not going to get into right now, you can analyze this data, you see these interesting pauses at certain places, okay? So for example, an experiment you could do, you can give it a designer DNA, okay? And if you want to understand where does RNA polymerase pause when it is reading through DNA, what nucleotide sequence, you can do it like this, right? Because you know if you have given a DNA sequence so much downstream, how much time until that will be reached. And I don't know how else you could figure it out, okay? So when does RNA polymerase pause? You can figure it out by these kind of experiments, okay? Right. So the last thing that I'm going to go. So remember that for us to actually start measuring things, you need to know this K. Okay, it is the stiffness of that spring, and there are different ways of measuring it. So that is called calibrating the optical trap. Okay, one simple way is to just use viscous drag. Okay. So what you do is that you trap an object, you keep it in the optical trap, and so here is let's say here is the cover slip and the laser beam is coming down from below, and here is the focus of the laser beam, and you have trapped this object here, okay? Now what I do, the trap position remains the same, but I move the entire flow cell to one side, okay, keeping this same. So, and I know the velocity with which I am moving it, and if it's a spherical object of a known size, then I can calculate the Stokes coefficient. And when I move this, I know that if I moved it at so-and-so velocity, I should have exerted so-and-so force on it, okay? 6 pi eta A. Of course, there are corrections because if you are close to the cover slip, you have to, you have to worry about all that, but let's not get into that right now. So this is what the data looks like. If this is the velocity pattern, okay, I am moving the stage with a certain velocity and with a constant velocity, but in order to move a certain distance, I have to move it for a certain time. Okay, let's say I'm moving it at 10 microns per second. Okay, and I'm moving it for 1 second, then I will move it over 10 microns. So, and so I gave it a certain velocity, and then I have to go back where I came from. And then I move it again, let's say in the opposite direction, then I stop here. Oh, sorry, this is the velocity plot. So I just stop here. So the velocity drops immediately to 0. I don't do anything for a certain time. Then I move it in the other direction. So I'm just doing this, okay? And when I do that, when you have a finite velocity, during that time, the bead will be displaced by a certain distance from the center of the trap, and I can simply say, I can equate these two, right? Because I know I know everything here. I know eta, because let's say I'm doing it in water, I know the viscosity of water, I know the size of this bead, sorry, that's the radius, and I know the velocity with which I moved that. Typically, the way this is done is that this whole thing is mounted on a piezoelectric stage, and the piezoelectric stage can move with a designed velocity depending on what current you are providing it. So you now calculate that if I have a velocity of so and so, I have a displacement of so and so. What is that? That is this distance, simply, okay? And I'm able to measure it because I'm projecting this onto that quadrant detector. And I just put in the numbers and I calculate K. Okay, once I have done this, I can move on to using a bead coated with a motor, okay, or whatever other experiments or RNA polymerase, etc. So that's one way of doing it, but this requires a very expensive piezoelectric stage. Okay, that piezoelectric stage could cost $30,000-$40,000, and you may have it or you may not have it. So there are other ways of also doing it. Right. So sorry, before I go there, how do you show that this spring is a linear spring? Okay, so one way to do that is simply increase the velocity. If you increase the velocity, then the displacement should increase linearly, okay, if it isn't still in the linear regime, and that's what you see here, okay, at least for this trap, okay. If you go beyond a certain velocity, then you'll go into the anharmonic region and it will not work, but yeah, so this is one simple way to show that it works like a linear spring over certain distances. Typically, that distance if you have a micron-sized object, that distance over which this spring is linear is 150, 160 nanometers. Okay. Another way to measure the stiffness of the spring, this optical tweezer spring, is that you don't use any piezo stage, nothing. You just keep the object trapped and you let it do its fluctuations. Okay, and these fluctuations are happening at a temperature which you know, and you will record. So this is time and this is the position. So you will record these fluctuations. Okay, you take the Fourier transform of this, and you now get the frequency component. So what are you plotting on the y-axis? You are plotting the variance at a certain frequency. That is called the power. Okay, at that frequency, you are plotting the variance, the fluctuations, amplitude of the fluctuations away from the mean, the squared amplitude at a certain frequency. And what happens is that, let's look at this, for example. Up to a certain frequency, there is a corner frequency. This is called, these fluctuations die out at high frequency, because those higher frequencies require larger energies. Okay, to move fast, you require larger energies, so it drops rapidly, and you can write the differential equation for this object and solve it, and you will get a Lorentzian. Okay, so this is the power, which is the variance as a function of frequency. This quantity on this side, this is thermal energy. This beta is 6 pi eta AV. Okay, and you fit it to this, and you can get this corner frequency, and from this, by doing various things, you can calculate the strap stiffness. Okay, which is there in this number here, but I won't go into that details. The other interesting thing is that this is nice to see, because if you are really in the center of a symmetric trap, the fluctuations in position should be normal about the central position. Okay, and so if I plot the histogram of positions about the center, you see really this nice normal distributions. Okay, Gaussian distributions. That means it is at the center, and because it's normally distributed, that you can understand that there are random fluctuations, and then you go into central limit theorem and all that. But so the other way, the last I'm going to wind up now, is again use the same kind of principle, that you measure these fluctuations. Okay, and you assume that these are partitioned equally in the different directions, and you simply equate the energy along a particular direction to the the energy from the spring in that direction, half kx square. Okay, that's assuming that force is equal to kx, the energy will be half kx square, and you equate these, and there you are now going to. So this is your trap stiffness, and this quantity you are measuring because this fluctuation. You can measure the variance. Okay, but typically when you do this, what you need to do is that you in certain situations, you need to do some kind of an anti-alias filtering of your data, okay, which has more technical details. I'm not going to get into that right now. Right, so there are things that you might, if you want to build fancier versions of this, you can have a motorized piezo stage, which is, I think, which is quite required for many of these measurements. You can have fluorescence with the optical trapping, which we tried to implement on our microscope, but we have it, but we don't use it. You can use temperature control. You can use acousto-optic deflectors for generating optical traps with constant load. Some of you can go and read why this is required. Fast detection of object displacement, that's the coordinate photodetector that I talked about. You can build versatile traps where you can trap many different objects together in. So what you can do is that you have a single laser beam, and you build a hologram. Okay, so you distribute this laser beam into multiple laser beams, and using the holographic device, you can manipulate the positions of these beams, and you can trap, let's say, one kind of organelle, another kind of organelle, and bring them together and engineer chemical reactions. So that's membrane fusion. You can do that. Now, this is the last slide. I want to talk briefly about something that we had done a little while ago. problem with a lot of these measurements is that you are always using an artificial cargo. Okay, you are using a bead, you know precisely the size of it, and you need to know the precise size to calibrate the optical trap. Okay, otherwise you can't calibrate the optical trap because you know all of those equations have a size component. Yeah. We figured out a way. Of course, we are not doing this inside cells. If I pull out an organelle from a cell, I don't know its size. Okay, it can be a certain range. How do I calibrate the optical trap for an object of unknown size? This has been a major problem. That's why people do most of these experiments using simple beads, okay, because they know the size. We wanted to do these kind of measurements, not just on beads, we can do that also, but on the lipid droplets that I talked to you yesterday about, okay. We saw those lipid droplets moving along those microtubules, remember, in vitro, and we wanted to measure what kind of motors, what kind of forces are there. So we did a combination of some of these things. We acquired, so we know that these lipid droplets are driven by a certain motor, okay. So you can get this kind of data, which is what I had showed it to you, okay. Now you generate this kind of stalls, but you measure these stalls by a video, which is by a camera simply, okay, and also in a quadrant photodetector. Remember that in a quadrant photodetector, you can measure the displacements much faster. But in a camera, you cannot measure it that fast because the camera is slower, okay. However, when this object stalls here at a horizontal region, it is a long duration. So you can, you don't need that kind of high time resolution. This plateau position, you can measure equally well with the video or with the quadrant detector. Is that understood? Right? Because it's a slow process. So you can measure it very nicely with the video, and we do that. Now, once you have done that, when you are measuring the displacement of this object in a quadrant detector, what you measure in the quadrant detector depends on the size of the object that you have tracked. Okay, because you can imagine it's a lens, right? So if the lens is larger, what you get on the other side will be larger. So this size, if it is a bead, you know it precisely, you can calibrate it. But if I trap a lipid droplet now and I trap another one, it is going to be very, very different, okay? How do I correct for this, okay? So what we did is that we did these kind of measurements on lipid droplets, and I now simply match this data with this data. I am using a certain unknown calibration factor. I'm assuming some calibration factor for the quadrant detector, which is wrong here in this case, because this is the real displacement. I simply recalibrate it using this data, okay? So now I know the calibration factor of my quadrant detector for a lipid droplet of an unknown size. I don't need to know the size, okay? Once I know that, then after all this is complete, I simply keep that object trapped in the lipid droplet, the lipid droplet trapped in the optical trap, and record data like this. Like this, okay? Now when I record this data, this is the position. I can find out the correct position because I have measured that using the video method, okay? I could not have done that otherwise. And once I have corrected for that, then I know the position, the correct position, and I know the variance, the correct variance. And from that I can get the trap stiffness for this lipid droplet, and that's what we did in this paper. If you people are interested, you can go and look at it. It's a sort of a technique development paper, and it's published. And I basically explained this to you. So this is what it looks like after it is corrected. Okay, and then there's another step where I just simply measure those fluctuations and recalibrate it knowing this information, and I can calibrate the trap. Okay, so all right, I think I don't have anything more. I will stop here.
All right. Questions?
Yes, please. I can hear you. Go ahead.
Yesterday you showed this lipid droplet experiment, where you showed that this lipid droplet is going away from the optical trap because it is having some maximum force, 20 piconewton.
Right.
So is it because this QPD means the size of the QPD that you talked today?
No, it is going away from the center of the optical trap because the motor is driving it.
Yeah.
Right? And then I can measure that displacement in the QPD.
Mhm.
But to exactly calibrate my quadrant photodetector, I need to know the exact size of the lipid droplet, okay, which I don't know. It can be, 30, 40 nanometers here and there, I have no idea. So I want to correct for that by using this method.
So how do you get the maximum force means of the optical?
Because I can measure that place where it is horizontal.
Okay.
And if I know the trap stiffness, which I will calculate in the next step from the fluctuations, I can go back and recalibrate this and get it.
Okay.
And we actually, when we did this, we found a force of 6 piconewtons for kinesin, which matches with what is known for beads. But that had never been measured in this way on quote unquote real cargoes. A bead is not a real cargo.
Any other question? Okay, if not, then let's thank Roop once again for a very interesting talk on motors and traps.
Next time you have to do this.